taking some time out of your day to join David and I on this webinar for characterizing fragile protein complexes. To begin, I'm just pulling up my agenda here if it wants to move. There we go. So to begin, I'll be introducing our featured presenter, David Dye, who will be presenting for the first part of the webinar, his case study on the observation of tuberous sclerosis protein complex polymerization from mammalian brains by electron microscopy. So we'll be looking at, what is pTSC and why study it, how David designed and purified an alpha TSC2 fab, and how he was able to actually purify for the endogenous pTSC as well. Next up, I will be taking over for part 2, and I'll be discussing the proteomics used for protein purification and characterization, and going through a couple of our services and how they were helpful and applied to David, as well as for general characterization purposes as well. So our featured presenter, David, attained his BSc from McGill University, majoring in immunology. And he did this in 2019 before joining the Jafari Lab in the Department of Medical and Biophysics at the University of Toronto as a direct entry PhD student. David is now a fourth-year PhD candidate whose research focuses on purifying and determining the atomic structures of human signaling proteins relevant to cancer using electron cryo-microscopy. For his efforts in his doctoral studies so far, David has received the Ontario Graduate Scholarship in 2020, as well as 2021, and the NSERC Canada Graduate Scholarship doctoral program in 2022. David's research has been published in multiple peer-reviewed journals, with the focus of his talk today having been featured in the ACS Biochemistry. So I am personally very much looking forward to this case study, and David, I will now pass it over to you.
Great, thanks a lot. I'll just share my screen and make sure everyone can see it. Is that visible to you, Monique?
Yeah, we can see it.
All right, great. I'll just get started then. Thank you, Monique, for that kind introduction, and thank you all for attending. Today, I'll be giving a presentation on my structural studies of the tuberous sclerosis protein complex purified from mammalian brains. But before talking about that protein complex, it's necessary to introduce a separate, equally important protein complex, the mammalian target of rapamycin complex 1, or mTORC1 for short. And what we need to know about mTORC1 is that it's a highly conserved signaling complex that promotes protein synthesis inside eukaryotic cells. And because it's at the heart of regulating such a fundamental cellular process, it itself finds itself regulating other important cellular processes that occur downstream of protein synthesis, such as metabolism, growth, proliferation. And for all these reasons, combined mTORC1 activity is itself meticulously regulated, which is somewhat captured in this complicated but nice figure of the mTORC1 regulatory and activation pathway. And the main takeaway from this figure is to understand that multiple signaling inputs converge in order to activate mTORC1 activity in response to all sorts of stimuli, such as glucose, or TNF, such as cytokines and growth factors. And these all inform about the internal and external energy and survival status of the cell to allow it to know whether it's a good condition or environment it's in to really push for protein synthesis and grow and proliferate in a healthy manner. And the thing to notice about all these signaling axes is that they all converge at a node at the center here in red. And this node represents the tuberous sclerosis protein complex, the focus of my structural studies. pTSC for short. So pTSC is a signaling hub that serves as the primary negative regulator of mTORC1 activity. I've shown here, highlighted in green, just captured from the previous figure, its canonical functional pathway. So normally what happens is that a growth factor will bind a cell surface receptor and causes the activation of the PI3 kinase, which phosphorylates this lipid called PIP2 into PIP3, and PIP3 activates PDK1, which activates AKT, which inhibits pTSC. And by inhibiting pTSC, it relieves its inhibition on mTORC1, thereby activating downstream mTORC1 activity and its associated processes such as cellular growth, proliferation, and survival. So for that reason, it follows that mutations in pTSC lead to a syndrome known as tuberous sclerosis complex disease, which is characterized by the manifestation of benign tumors across the body, seemingly indiscriminately targeting all sorts of organ systems. And really what this is a symptom of is just unregulated cellular growth as a result of dysregulated mTORC1 activity. So it has a very heterogeneous disease profile depending on where such benign tumors target. So for instance, if it ends up in the heart, you get some cardiac diseases and syndromes. If it ends up in the skin, you get this weird patchiness of your skin. Or perhaps the worst place it can end up, these benign tumors, are in the brain, where oftentimes the prognosis is fatal. It leads to severe seizures, severe developmental delays, autism spectrum disorders. And for this reason, it's actually quite a serious disease. Luckily, it's rare. It affects 1 in 5,000 globally, but due to how heterogeneous it is, it's very difficult to treat. And so far, there is actually no singular effective treatment for TSC disease. That is changing a little bit now that we understand the structure and the composition of the pTSC a lot better than we did even just 5 years ago. So recent cryo-EM structures of recombinant pTSC that was overexpressed in mammalian cells and reconstituted in vitro have shown that it's a very beautiful molecule. It is in the shape of a banana, and it's composed of 3 unique subunits, TSC2, of which it has 2 subunits, TSC1, which it again has 2 subunits, and this one accessory protein here shown in pink, just off in the side, called TBC1D7. And here are just the cryo-EM structure of the core of pTSC as determined by Yang et al. In 2021. And it, as I just mentioned, reveals its overall architecture and its subunit stoichiometries. In addition, during this flood of cryo-EM information about the pTSC complex in 2021, other groups, Ramlal et al., published their findings that pTSC has this polymeric potential to oligomerize into extended filaments. And this was really on the backs of both direct and indirect evidence. Some of the direct evidence were just direct cryo-EM observations of this protein. If you can follow these very light yellow dashed lines of these proteins simply forming these like head to tail assemblies, which they diagrammatically show here as a possible proposed oligomeric mechanism. And this is a cryo-EM micrograph, I should mention, where in black you have darker protein against a whiter and lighter background of flash frozen crystal clear ice. So that's some of the direct evidence of the polymerization of pTSC. There's also these indirect lines of evidence. And one of them is simply this arch shape. Describes a very gradual curve of the overall architecture of the protein. It seemingly suggests that this protein likes to hug curved surfaces, and where else than lipid membranes and phospholipid bilayers. So it seems to suggest this ability to lace these membranes, which is consistent with the active localization of pTSC, which is at the outer lysosomal membrane, and it is in this compartment that it exerts its inhibitory effect on mTORC1 activity. And this is actually a model proposed by Yang et al., where this is supposed to represent a lysosomal membrane, and then the shape of the membrane kind of induces the head to tail polymerization of the arch shape of pTSC on the peripheral outer surface. And then there's another line of indirect evidence that I don't show here, but is worth mentioning, and it's that it's not uncommon for these signaling proteins to undergo some kind of oligomeric transformation in order to regulate their signaling ability. So by forming these higher order oligomers, either the signaling potential of that molecule is amplified in some manner, or sometimes it's also inhibited by storing it in a higher order stable reserve of protein until conditions are right for it to be functionally active again. So all these lines of evidence kind of suggested that there's something higher order going on when it came to pTSC. So to that end, the motivation for our studies of pTSC occur from the fact that, as I just mentioned, all these experimental structures of pTSC to date are recombinant, meaning they were overexpressed in mammalian cell culture, purified, and then reconstituted in vitro. And even the best one, which is the one I was showing very detailedly, this one is 30% complete. So, and it only really describes the core components of pTSC, and only those components that we thought to add to our reaction mixture. None of it doesn't capture perhaps the unknown protein interactions pTSC makes endogenously in situ or in vivo. So to that end, there is actually not a complete atomic structure of pTSC, and this is certainly hampering efforts in developing effective drugs and treatments against this very challenging TSC disease. And the purification of endogenous pTSC could serve as a logical approach to ameliorating these two just mentioned challenges, because an endogenous pTSC, so one purified directly from cells, not overexpressed in cells, then reconstituted in a test tube, but instead one directly purified from native sources, could preserve these important post-translational modifications that are highly clustered on important signaling proteins, and also could preserve as of yet unknown protein interactions that pTSC may be forming. And the combination of these factors could be crucial in stabilizing the full-length structure for structural determination and or explaining this polymeric behavior that's been hinted at. So to that end, the goal of this research was to purify an endogenous pTSC from the organ we chose was brain tissue, because this is an organ in which pTSC is naturally enriched. And with these endogenous samples, study them structurally using methods in electron microscopy. And to complete this, there were three aims necessary. The first was to actually engineer a protein capable of purifying pTSC from mammalian tissue, because unlike in these other systems where you can either just give them a plasmid with a tag, or engineer the cell line itself to give itself an endogenous tag. If you're purifying from something like organ tissue, you obviously can't easily edit the genome of this to tag your protein. So most often what you need is a separate protein that is highly specific for the endogenous target you're interested in purifying. So this was necessary to establish and to purify and demonstrate the functionality of. And then once we had such a protein, we wanted to purify endogenous pTSC, and then we wanted to study them using electron microscopes. So with regards to aim one, the design of a recombinant alpha TSC2 Fab protein, meaning a Fab protein specific against the TSC2 subunit of the pTSC complex, we underwent this pipeline in order to obtain such a protein. So there is this very literature validated anti-TSC2 full antibody from cell signaling technology. It's demonstrated in multiple high impact and highly cited papers that it has a great ability to IP pTSC, or rather TSC2. So we obtained this antibody, and then we enlisted the services of Rapid Novor to obtain the sequence, the protein sequence of this antibody de novo. And from what I understand of Rapid Novor's pipeline, they essentially took our antibody, digested it with a cocktail of proteases, and then ran it through their LC-MS machines to obtain peptides. And then they were able to recombine these peptides that were contiguous with each other to create a de novo protein sequence that they assembled, and were able to then give us the precise amino acid sequence of both the heavy chain and the light chain of the antibody. Perhaps I should mention that the basic construction of a full antibody is that it's composed of two regions, an FC region and an FAB region, the Fab region. And the Fab region is that which is specific against specific proteins. And the regions are composed of light and heavy chains. And since we're interested in reconstructing the Fab region, we needed a full light chain and half a heavy chain. So these were the sequences Rapid Novor was able to synthesize and sequence for us. And then we, using these sequences, put them in our own plasmids under this type of construction where the heavy chain was C-terminally tagged with a 3X flag tag, which becomes important later. And additionally, the light chain was C-terminally tagged with a CBP tag for protein purification. Then there's also this N-terminal and sec translocase signal. And the importance of this domain is to ensure that as the protein is being translated, it's getting secreted out of the cell. And this makes the purification of the protein much easier. So describing the purification of the anti-TSC2 Fab, what we did is we took HEC293F cells, a mammalian cell culture of human embryonal kidney cells, and we co-transfected them with the two plasmids we were able to generate off of the information that we obtained with the help of Rapid Novor. And then we allowed these transfections to grow for 96 hours before pelleting them. And the key is that now we keep the supernatant, because with the sec translocase signal, each chain should have been secreted into the surrounding culture medium. And in that medium, the light and heavy chains should recombine to form a Fab. So with this supernatant, we pass it across our 1 mil calmodulin agarose column, a column specific against the C-terminal CBP tag of the light chain. And then once we collect some elution fractions there, we do a final gel filtration polishing step that separates proteins by size. And the goal is to get finally a purified preparation of the anti-TSC2 Fab. So going through all those steps, this is the result of the purification. The light chain has a theoretical molecular weight of 26.5, the heavy chain 25.9, and then so the recombined heterodimer should be around 50 kilodalton. So that's precisely what we see. This is a SDS page gel of the CBP column purification. And this enriched band here is are both the light and heavy chains. And it's important to recognize that this is both the light and heavy chains because they run at the same size. And this SDS gel is denaturing, and it is also, the sample loaded onto it has beta-mercaptoethanol, which reduces disulfide bonds within the structure of an antibody. So it separates physically the light and heavy chains such that they now run as separate proteins. And for that reason, they run on top of each other. But to make sure that we are in fact purifying an intact heterodimer, which is impossible to tell from a denaturing assay, I perform a non-denaturing assay as well, or not non-denaturing, but not non-reducing assay as well, in which I run Fab that I purified in the presence and absence of beta-mercaptoethanol. And what you see is the size shift you would expect, where if the disulfide bond is preserved, it runs at its intact 50 kilodalton weight, whereas if it's not preserved, they once again run as separate chains, which overlap on each other and appear as just above 25 kilodalton. So based on these results, we're quite confident that we expressed, purified, and successfully designed this anti-TSC2 Fab protein. And the next step was actually to then demonstrate the functionality and specificity of this Fab for its intended target, the TSC2 subunit of pTSC. Yeah, so that is what I just said. And the way we wanted to test its specificity was by directly seeing if it could IP or target TSC2 from subcellular fractions prepared from mammalian brain tissue. So the brain tissues we targeted were either from rat or from a more complicated mammalian brain, such as pig. And the process of subcellularly fractionating them was as follows. We homogenized them, and then we spun the homogenate to obtain a supernatant 1 fraction and a pellet 1 fraction. The pellet 1 fraction is uninteresting. It just contains whole cell debris. So we further processed the supernatant 1 fraction by ultracentrifugation to obtain a supernatant 2 fraction and a pellet 2 fraction. So both these fractions are actually interesting. They both contain populations of pTSC, but the one we're more interested in studying was the one inside the membrane pellet fraction, because as I mentioned before, pTSC is targeted to the lysosomal membrane in its active form where it inhibits mTORC1. And as a peripheral membrane protein, we should expect to see its enrichment in the membrane fraction. So we solubilize the membrane pellet using a non-ionic detergent called endodecyl multicide, DDM for short. And what this does is just it solubilizes the membranes and those proteins integral and peripheral on the membrane, so that after another homogenization step and a final high-speed spin, we obtain this S3 supernatant 3 fraction that should contain a solubilized population of membrane proteins, in theory enriched with the pTSC we're trying to purify. So having done this subcellular fractionation protocol, we can then imagine a Western blot assay that precisely tests the specificity of our alpha TSC2 Fab. So if we collect these S1, S2, S3 fractions across rat and pig brain, we can then run it on a gel, transfer it onto a membrane such that all the proteins inside those supernatant fractions get adsorbed onto a nitrocellulose membrane. And as a simplification, I show specifically a pTSC complex adsorbed to this membrane. And in theory, we could then introduce our Fab as a primary antibody, which binds to the C terminus of one of the subunits of TSC2. And then we can introduce an alpha M2 antibody as a secondary antibody. And alpha M2 anti-M2 is just a commercial antibody raised against 3X flag tags. So it would bind the C-terminal 3X flag tag of the heavy chain that we specifically engineered into the Fab. And finally, we can introduce the standard tertiary goat anti-mouse HRP conjugated antibody that allows the ECL chemiluminescent reaction to occur that enables visualization of a Western blot signal. And what we see is a very clean signal for the TSC2 subunit running at its correct molecular weight, around 200 kilodalton. And we see after normalization, certainly a great enrichment of protein and S3. So this is exactly what we wanted to see. And we're confident at this point that the Western blot confirmed that the Fab was indeed specific and functional. So we're now prepared to attempt a purification of pTSC from mammalian brain tissue. And the protocol looks exactly as the subcellular fractionation. However, now with S3, we pass it over in alpha M2 agarose column. So recall the M2 antibody is specific for the flag tag, and now we're leveraging its specificity for the flag tag towards protein purification by packing a column full of M2 agarose beads, which are just agarose beads conjugated covalently to M2 antibodies, such that you can imagine after you pass through S3 into such a column, what's going on molecularly is that these beads will sequester and capture Fab, or an important first step is that the column is pre-charged with Fab, I should mention. So even before the S3 is passed over the column, we pass over the Fab in order to charge all of these open Fab spots on the M2 antibody with Fab. And then once you introduce S3, pTSC floating around in S3 will get specifically bound by the Fab in a similar manner to how I demonstrated in the Western blot. So this is the binding step. And in order to then elute the complex of pTSC bound to the Fab, you introduce synthetic 3X flag peptides, which I just show as these squiggly lines, so that these peptides compete for binding to the M2 antibodies and competitively elute a pTSC Fab complex. So in order to validate that it works, we performed Western blot on a purified sample, and I will just explain the loading of this Western blot on the left, which is then applied to the two other Western blots. So these fractions here are just S3. They serve as positive controls to show that indeed the subunit of TSC we're blotting against was present in the lysate. And in this case, we're blotting against TSC2. And these are the elution fractions off the flag column. And as you can see, we see the preservation and the purification of this TSC2 subunit, which was great to see because that was the subunit against which our Fab was designed. This prominent signal down here is important to mention as well, because you will see it across all my blots. This is actually the Fab. And this is not surprising because the Fab itself is based on a TSC2 antibody that was raised in rabbit. And we're using a secondary antibody that is specific against rabbit. So we should expect our antibody, our Fab, to also give a secondary signal upon using a goat anti-rabbit HRP conjugated antibody. So we clearly see the purification of TSC2 from our elution fractions. And importantly, we see no purification when simultaneous to the purification, I have a run where I don't charge the column. So this is to test non-specific binding of the pTSC complex to flag agarose speeds. And we see that purification of TSC2 subunit occurs in a Fab-dependent manner. This is recapitulated for TSC1, and then also the TBC1D7 subunit. And a fair observation would be, how do you know that these bands down here are not just the Fab? Well, in this case, we changed the assay a little to make sure we don't get any overlapping signal from the Fab. The Fab is, of course, engineered from the variable region of the antibody, and clearly the secondary antibody we were using before was binding the variable region of the antibody. But some secondary antibodies have been engineered to be only specific against the FC portion of antibodies. So that is what we used here. We used a secondary goat anti-rabbit FC specific HRP conjugated antibody, such that this secondary antibody would only bind to FC regions, therefore only the TBC1D7 antibody, and not the Fab and not the Fab that we use to purify the protein. So we can be confident that this signal is derived from only TBC1D7 in that case. So this was very nice to see. We saw very good purification, or we saw purification of endogenous pTSC from rat brain tissue for the first time, and in a Fab-dependent manner, very importantly. So we repeated the same type of analysis and purification now on pig brains, and it was loaded similarly. And just walking through the blots, this is the blot against the TSC2 subunit, and we see this characteristic TSC2 band. I'll talk about the TSC1 blot after I first discussed that we also saw the purification of the TBC1D7 subunit using this similar anti-FC secondary antibody trick. And when we probed for TSC1, initially we saw nothing. So when you do a rabbit anti-TSC1 Western blot against the TSC1 subunit from our purified fractions, you don't see the characteristic band that we expect to see around 180. And when I looked into it, I found that pig TSC1 and rat TSC1 have high sequence variability in the region that the rabbit anti-TSC1 antibody is predicted to bind the pig version. And obviously this can be very bad for antibodies, which depend highly on specific epitopes for highly specific binding. So we went out and purchased a mouse anti-TSC1 antibody that bound in a different location that was more conserved across pig and brain. And what we found was indeed we were able to recover a TSC1 signal in addition to some other bands. And these bands here, once again, are the Fab and the intact and some. This is not the intact Fab. This is some kind of secondary contamination that is always seen when using this mouse anti-TSC1 antibody. But in any case, we're able to prove also the purification of all the subunits of the core pTSC complex from pig brain tissue. And that was also very nice to see. So now that we had these purified pTSC samples, the next thing we wanted to do as electron microscopists was, of course, to look at it by electron microscopy using a method called negative stain electron microscopy. So for those of you that don't know how the method works, just a brief overview is that we start with something called an electron microscopy grid. This is just a circular metal mesh, approximately 3 millimeter in diameter. And every EM experiment occurs on a grid. And it's the vehicle that loads our samples into the electron microscope for observation. So we take this grid and let's just flip it 90 degrees into the plane of the slide. And then what we do is we apply a layer of amorphous carbon onto the surface of this grid, such that you can imagine now there's this gray layer of carbon. And the point of applying this carbon is that it provides a solid support onto which, here I just show a generic protein, can be adsorbed onto the surface of this carbon, such that when you blot away the excess protein solution, what you keep is this immobilized population of protein. And then you can go ahead and stain it with a heavy metal stain. In this case, this is the uranyl acetate. It has this very heavy uranium atom, and this becomes important later, such that now you stain your protein, and then you plot away the excess stain. And the end result you can imagine is, if you were to zoom in on one of these proteins, what you can imagine is a setup like this, where you have your protein embedded in a thickness of heavy metal stain all atop a thin layer of carbon. And now if you were to image this setup under an electron microscope, so in a source of electrons, and each electron here is just shown as an individual sine wave, what happens is that in places where there is only stain, less electrons are able to transmit through to the detector of the microscope, because as compared to a place where electron hits protein, which physically extrudes heavy metal stain in that location, more electrons are able to transmit through and hit the detector. And that's just because proteins happen to be made of more less heavy atoms, such as carbon, hydrogen, nitrogen, oxygen, sulfur, phosphorus, these types of lighter atoms as compared to something relatively gigantic, such as uranium. So you get more electron transmission in places where there is protein, and the end result is a negative image in which the protein is white against a darker background. And here I show a example model protein, which we work very closely within the lab, the fatty acid synthase from fungus. So now understanding the basic methodology of this technique, we applied it to our purified samples of pTSC, and we saw these consistent pattern of these rod-shaped formations. And these rod-shaped formations are consistent with the cryo-EM studies in dimension that I discussed previously. So we were pleased to see that we were likely purifying an endogenous fraction of pTSC, and this is from rat brain. From pig brain, the results were even clearer, where these rods were very enriched as compared and very obvious and visually striking. You can even sometimes see perhaps the polymerization in action, where you see these bends in these rods and the characteristic arc shape sometimes really being visually obvious. But perhaps the most striking thing we saw from pig brains was this large scale oligomerization into just these, relatively speaking, gigantic strands that just spread across the entire field of view of the microscope. And these were very striking to see at the time, because it kind of is almost a direct observation of what had been hinted at in the literature, and at a resolution where you can sometimes even see the cross-section, the interface of these head-to-tail oligomeric interactions, and the higher order bundling and aggregation into these aggregates of pTSC purified from pig brain. So this was a very nice thing to observe, unique to an endogenous system that had simply been overlooked or perhaps unable to be captured by in vitro systems for whatever reason, perhaps due to some of those reasons I talked about at the start of the talk, such as post-translational modifications and things of that sort. So lastly, we just wanted to confirm that this was indeed pTSC, and to do that, we did a method called immunogold labeling electron microscopy, iGEM for short. And it's very similar to negative stain. It's all the same theory, but now I've conjugated the protein on a grid to my fab, and an antibody specific against the flag tag conjugated to a gold particle. And the gold particle is obviously a heavy nucleus, so that anytime an electron hits the gold particle, there's zero transmission, or very nearly zero transmission onto the electron detector. And what this results in is a black dot in the microscope. So everywhere where you see a black dot is somewhere where the electron encountered a gold atom, and the gold atom should be physically proximal to the protein of interest because it is tethered to it by an antibody connection. So once again, this is a fungal fatty acid synthase. And when you perform this type of analysis on our purified pTSC from rat brains, what you see are the formation of these dots into these longer layers, into these aggregates, and they kind of recapitulate this rod-like shape that we observed, both in cryo-EM, both in recombinant studies, and of course our own endogenous purifications by negative stain electron microscopy. And it was nice to also see them repeated in pig brain. So the combination of our Western blot data, our purification data, our negative stain data, and finally our iGEM data really presented a nice holistic argument that we indeed did purify endogenous pTSC and observed something very cool about it in an endogenous system. So to wrap up, what we did in this study is we demonstrated really the power of Fab protein sequencing technology as a novel method of purifying challenging and fragile protein complexes. We validated and performed the first purification of endogenous pTSC and found that its shape was consistent with those seen in recent cryo-EM studies of its recombinant version. And then we did a native purification of pTSC and observed this unique oligomeric tendency and ability of it that was slightly overlooked in the recombinant studies. And we found that purification of endogenous proteins in general moving forward could allow us to study more of their biologically relevant native states and could reveal novel biology about these proteins. So looking forward, we're interested in doing cryo-EM of pTSC filaments to elucidate the structural basis of its oligomerization. And finally, to apply fat-based purification on other challenging endogenous protein complexes. So with that, I'd like to thank you all for listening and like to thank all the members of my lab. And that's the end of my presentation. So thanks a lot. Monique, I'll hand it back to you, I guess.
Yeah, thanks, David. Let me just get my screen going here. Okay. So thanks again, David, for that really insightful and exciting presentation. From my end, it's always great to learn about how our services can help contribute to research like yours, and definitely looking forward to working with you for future projects. So now we'll go into the second part of our webinar, where I'll be detailing how our various services and applications in proteomics can be used to purify and characterize fragile protein complexes. Not going forward. Here we go. Okay. So I put together a list of rapid Nova services. We do have a couple that are centered around the use of next generation protein sequencing and others that are more applicable for quality assurance and characterization. But for the purposes of this presentation, I'd like to focus on the ones that I've highlighted for you here. So we have monoclonal antibody sequencing, which is REMAB, as well as our HDX-MS epitope mapping service and our SPR kinetic analysis service. Now, I'm not going to spend too much time on this slide, and I will come back to it. But I just want to highlight that the services I'll be discussing in tandem with some key points about general purification and characterization, and of course, that ones that were related to David's work. So the core technology that we have is really the combination of bioinformatics, proteomics, mass spec, and machine learning algorithms. And we combine all of these three things together to provide insights and results and analysis for our clients and customers to help them move forward with their research. So the first service that I had mentioned is REMAB. So it's our next generation protein sequencing service. David alluded to this a little bit earlier in his presentation as well, but I'll just give you a bit more detail. Generally, we'd be working with a purified monoclonal antibody that we would then run between 5 to 10 different enzymes on, or as David mentioned. So when we're doing our sequencing work and we're happy with the data, we put everything together in a nice final report for you. So it would detail, as David mentioned, the full length sequences of the antibody. So we have our heavy chain, we have got our light chain. We also have our variable regions, so CDRs as well as the constant region. We're also able to pull out some post-translational modifications. So if there's any weird cleaving in the N-terminus or C-terminus, or if there's N-linked or O-linked glycosylation, then these are things that we'd be able to let you know about. The requirements for the service are quite simple. We ask for 100 micrograms of purified antibody. Now, because we are working with mass spec, it is important that we work with pure samples. So protein contaminants that are picked up by the mass spec can make it difficult to obtain good quality data. And we want to avoid as much background noise as possible. So what's so special about what Rapid Nova does? Well, we have industry-leading quality standards, and we achieve this in a few different ways. So first off, with the depth of our coverage, we aim for 30-fold coverage in all of our sequencing results. So 30-fold coverage over every single amino acid. And that means that we get to a confidence level of 99.9%. Additionally, we also have the ability to differentiate between isobaric amino acids, isoleucine and leucine, using our wild technology. It isn't just wild because it's crazy. It's W-ion isoleucine leucine differentiation. So typically these two amino acids can be difficult to differentiate because they have the same methods. But this technology is applied in all of our monoclonal sequencing projects, and it's especially important when looking at point mutations or targeting specific epitopes like David did. Additionally, as I mentioned earlier, we do have independent reviews. So we're always going to be looking at the data with at least two different bioinformaticians as well. And we don't accept less than 100% accuracy in our work. It's one of the reasons why we have the requirements that we do and why we've set up really strong quality controls throughout our sequencing process. So in regards to epitope mapping, we use HDX-MF, which measures the exposure of hydrogen molecules on the surface of a protein or protein complex after quenching with deuterium. So how this is done is you would incubate different fractions with D2O solution in a time series. And then through quenching, you'd control temperature to prevent further exchange or back exchange. The complex is then digested and run through our mass spec. Finally, the results from the mass spec would be analyzed and plotted. Now, this is done for both the bound as well as the unbound complexes. So hydrogen molecules that are buried within the structure, for instance, at the binding site of an antibody antigen complex, they wouldn't be exchanged as readily. And by examining the intact proteins and proteins via mass spec and comparing them with the amino acid sequence, it's then possible to determine which regions were engaged in binding. In regards to benefits, there are a few, but a key area I'd like to highlight here is that we can look at linear conformational and structural epitopes. So when you're looking at protein complexes that are potentially fragile, this might be something that would be of more interest to you to understand different conformations of the protein and what areas are engaged in binding itself. In regards to applications, as I mentioned, there are quite a few here, but I'd like to highlight the two listed there in bold. So pertaining to David's work, antibody antigen studies through HGMX are a good way to test whether areas of the native proteins are binding to the Fab. In cases similar to David's, maybe you'd want to look at a variety of antibodies that bind to different epitopes of a complex protein. For antibody engineering studies, understanding what areas of your proteins are involved in binding can help you engineer proteins maintaining key epitopes or perhaps changing them for lower affinity in some circumstances. In regard to SPR kinetic binding analysis, this is done by immobilizing a ligand to the metal surface of a sensor chip. And then we would be injecting an analyte at a specific flow rate and concentration. So once equilibrium is reached, a running buffer would then be continuously applied, and then the bound analyte would begin to disassociate. So this disassociation is measured by the detection system and then plotted to provide kinetic insight on the binding of the analyte and the ligand. Now, again, there are a few advantages to using SPR, but I'm going to highlight the ones in bold. So real-time monitoring is important here. The association and disassociation of molecules can be analyzed in real time. So we're able to provide on and off rate information in addition to affinity data. We also have label-free detection. So labeling of the ligand or analyte isn't required, which permits the natural functionality of the molecules and results in a more accurate depiction of the actual biomolecular interaction. And then in regards to low sample concentration intake, small sample volumes are required to do this work. So you don't have to generally make more. So less time spent for sample expression and purification and preparation here. So I promised we'd come back to the slide, and here we are. So to summarize, our next generation protein sequencing service, REMAB, can be useful when adapting a standard antibody to a targeted tool one for native protein purification, like we saw in David's case study. HDX, MS, and SPR are also useful characterization services to target potential tool molecules and can help make sure that the engineered antibody is consistent with what you expected or what you'd like for successful isolation of the fragile protein complex in regards to looking at different epitopes or affinities. If you want to take it further and look at different parts of the complex protein, potentially, we can also sequence native proteins with REMAB, and we can help you to test different tool molecules and understanding different protein complex binding and conformational changes as seen in HDX and SPR as well. So as the largest privately funded proteomics facility in Canada and having worked with over 6,000 different proteins and antibodies for sequencing or characterization, we are very committed to driving research forward like David's. And additionally, we're working towards our mission to elevate human health by decoding immunity. So that brings me to the end of our webinar. Thank you all for listening, and I'll open up the floor for any questions.
So I actually see a question from Angelo in the chat that I'll address now. What was the question specifically? So he asked how much purified protein is needed for EM and in what purity? So that's a great question. So EM is a very powerful technique, and that you don't need much purified protein at all to perform an interesting experiment. What really matters is the purity of that protein. And even then you can you have a little bit of leeway when it comes to the purification of the protein. The best way to describe the purified protein needs and the protein yields is to compare it to X-ray crystallography, which is a technique where you need very high yields of protein and extremely pure protein samples such that it crystallizes properly. In EM, this is not so much the case. And you saw in my presentation, the way I was assessing purification of pTSC was by Western blot, which is basically as sensitive a technique as you can get. And the reason for that is because there was not a lot of pTSC to begin with in the samples that I was purifying from. So I was purifying from brain tissue, especially for rat brain tissue, that's like only a couple grams. So already there, you're limited in your starting material. And that was actually the rationale to look into pig brain, because it's a brain almost 10 times as massive. But you're limited in yield. And so the main consideration then is the purification. And you want to put as pure a sample as possible onto an EM grid to get the most definitive and nice micrographs. And that's what we aimed for in the pTSC study. As soon as we established a solid purification scheme and an assay to validate it by Western blot, what we put on the grid is exactly what we saw. And in that case, you saw these rods consistent with previous studies. And then we went a step further by doing immunogold electron labeling, immunogold electron microscopy. And that was really what definitively convinced us that the purification worked. So I hope that answered your question. So I'll keep going through. There's a question from Kelly for me, I think, and then there's one for you, Monique. I'll just answer Kelly's really quickly. You state fragile proteins, but wonder how you characterize this in terms of the complex. Let me think about short half-life rapid dissociation. Yeah, so I think what you're trying to say is TSC actually a fragile protein complex? And these are some aspects of the literature I didn't get into out of the interest of time. And I think we can confidently say TSC is both a challenging and fragile protein complex because the cryo-EM structures I showed in my talk of the recombinant structure, there was a detail about its purification that I didn't mention, and it's that it was actually stabilized by a cross-linking molecule using a method called graphics. It involves taking your purified protein and then running it through a sucrose gradient by spinning it by ultracentrifugation in a gradient of denser and denser concentrations of glutaraldehyde, the chemistry of which I'm not too sure of, but I need to refresh myself rather. But it cross-links the protein and stabilizes its weak protein-protein interactions. So the fact that the only cryo-EM structure we have of pTSC was necessary to be processed in such a manner, which you can argue is very non-biological, goes to show that, yes, certainly the protein is fragile if you need artificial cross-linker to stabilize it. And the reason why I potentially saw rods in my negative stain preparations without any cross-linking is because I was looking at an endogenous fraction, and the endogenous fraction is biologically different. There are things going on in that endogenous fraction that certainly people that tried to purify it recombinantly just don't have any ideas going on, like proteins that they didn't even think to add to their test tubes. So my hypothesis is that these endogenous factors also played a role into stabilizing the rods and the long filaments I saw in my work. Yeah. So hope that answered your question.
All right, and there is a question from an anonymous attendee. So in terms of our need for recombinant protein sequencing, for antibodies, it's just the 100 micrograms in a standard buffer like PBS. And as I mentioned, we would want over 80% purity there. For non-antibody proteins, I would say let's have a conversation. Concentration and buffer can have an impact there. No, there is no size limit. We would just want to be working with a purified protein. Generally, 100 micrograms is safe. If it is a larger protein, then it is a possibility that we could ask for more. It can vary slightly case by case for non-antibody proteins. And I see one more about SDS page. So the question is, do you also accept SDS page gel cut proteins for sequencing? Yeah, we can work with that. We don't often get SDS page cut proteins, but it is something that we can take out of the gel and continue our sequencing process for. All right. It is now just a minute over the hour, and I don't see any more questions coming through. So I think we'll end it here. Thanks again, David, for joining me, and thank you everyone for tuning in.
Thank you.
Bye.
Bye.